Jai Hind friends. In this lecture, we will see the transcription process. We will see what is the meaning of the transcription process. Then we will see where it takes place, when it takes place, which enzymes are required for the transcription process, and then we will see what are the actual steps that are present in the transcription process. First, we will see the meaning of the transcription process. The meaning of transcription process, it is the process in which the information that is present or stored in the DNA strand, it is copied into the new molecule of the RNA with the help of RNA polymerase enzyme. This is the process of the transcription. DNA से RNA फॉर्म होना, इस प्रोसेस को क्या बोलते हैं? Transcription process. Then when the transcription process takes place, it takes place in the G1 and G2 phase of the cell cycle. G1 and G2 phase. Replication, it takes place in the S phase of the cell cycle, while the transcription process, it takes place in the G1 and G2 phase of the cell cycle. Then then the enzymes that in transcription process, in case of prokaryotes, only the single enzyme, that is the RNA polymerase, is required, while in case of the eukaryotes, there are mainly RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3 enzymes are required in case of the eukaryotes. In case of the prokaryotes, this single RNA polymerase enzyme, it will transcribe all types of the RNA, that is mRNA, that is ribosomal RNA, and the tRNA. While in case of the eukaryotes, RNA polymerase 1, it is responsible for the transcription of ribosomal RNA. Yes, RNA polymerase second, दोस्तों, it is responsible for the transcription of the messenger RNA, and third one is for the tRNA. But remember the one thing, RNA polymerase directly do not form the mRNA. It forms the precursor of the mRNA, that is heterogeneous nuclear RNA. Yes, RNA polymerase 2, it will form the first hnRNA and then the mRNA, while RNA polymerase 3, it will form first small nuclear RNA and then tRNA. First hnRNA, then mRNA. The steps in the transcription process are same in case of both prokaryotes as well as in the eukaryotes. First, there will be initiation, then elongation, and at last there will be the termination of the transcription process. Then we will see the transcription unit. The transcription unit is the part of the DNA that takes part in the transcription process. Actually, it is the segment of the DNA or part of the DNA that actually takes part in the transcription process. It consists of mainly 3 parts, that is the promoter region, the structural genes, and the terminator region. Now remember student, the new RNA formed will be in the direction of the 5' to the 3' direction. New RNA formed will be in the direction of the 5' to 3' direction. Means the polarity of the DNA strand which is acting as a template strand should be in which direction? 3' to 5' direction, 3' to the 5' direction. It will act as a template strand. Means this part of this segment or strand of the DNA, it will be the template strand whose direction is the 3' to the 5', and its complementary strand, it will be called as the coding strand. It will be called as the coding strand. Now student, pay attention here. In the replication process, both strands of the DNA takes part in the replication, but while in case of the transcription process, only the one strand of the DNA takes part. Now there are mainly 2 reasons why both strands of the DNA are not copied into the RNA, or both strands of the DNA do not get transcribed. If both strands act as a template, then 2 complementary strands of the RNA will be produced, and there will be pairing between the 2 complementary strands. कैसे देखते हैं? If adenine है तो mRNA का जो नया strand रहेगा उस पे कौन सा न्यूक्लियोटाइड रहेगी प्रेजेंट? यूरेसिल. यस, देन ग्वाइनिन है तो साइटोसिल, साइटोसिल है तो ग्वाइनिन, थाइमिन है तो एडेनिन, एडेनिन है तो यूरेसिल न्यूक्लियोटाइड प्रेजेंट रहेगी. अगर दूसरा भी स्ट्रैंड टेम्पलेट स्ट्रैंड करके एक्ट हो जाता है तो थाइमिन है तो एडेनिन, साइटोसिल है तो ग्वाइनिन, ग्वाइनिन है तो साइटोसिल. अगर डीएनए पे एडेनिन है तो जो आरएनए होगा उस पे क्या रहेगा प्रेजेंट? यूरेसिल, एंड थाइमिन है तो एडेनिन. अभी 2 strands है RNA के, these are produced if both strands act as a template strand. If both strand of the act as a template strand. Now look at these two strands. Both these strands, this strand, it will have direction of 5 prime to 3 prime, and the direction of this strand, it will be the 3 prime to the 5 prime. Now these two strands are again complementary, and they will form the double stranded RNA. And this double stranded RNA, it will prevent the RNA from being translated into the proteins. Means this RNA, it will not undergo the translation process and it will not form the proteins. This is one of the complication. Second complication, this is the segment of the DNA which is undergoing transcription process. Now this segment of the DNA, it is producing two different types of the RNA. It is producing the RNA having sequence UCGAU, and the another strand having sequence AGCUA. Now these two strands or these two RNA, they will code for the two different types of the proteins. They will code for the two different types of the proteins, and it will complicate the genetic information transfer machinery. Means same segment of the DNA is coding for the different types of the protein, which is again the complication for the genetic information transfer machinery. And this is again against the central dogma of the molecular biology. That's why only the one strand of the DNA is transcribed into the RNA and not two strands. So transcription process may only one strand takes part in the transcription process, and that is the template strand. Only one strand of the DNA acts as a template strand. There are only two reasons. Which are those? First, it will form the double stranded RNA and it will complicate the process of the translation. And second reason is the same segment of the DNA, it will code for the two different types of RNA, it will code for the two different types of the proteins. Means same segment of the DNA is coding for the two different types of the proteins. Again, it is against the central dogma of the molecular biology. This is the reason why only one strand of the DNA, it takes part in the transcription process and not both. Then we will see the promoter region, structural gene, and the terminator region. Now these are the part of the transcription unit. One transcription unit, it consists of the promoter, structural gene, and the termination. Now this, what is promoter region? This promoter region, it is the site which provides binding site for the RNA polymerase enzyme. Binding site for RNA polymerase enzyme. Yes, this promoter region, it is present at which end of the coding strand? Yes, it is present at the 5 prime end of the coding strand. 5. Now student, the transcription process, it takes place like this, from this 3 prime direction to the 5 prime direction of the template strand. That's why the transcription process starts from here, where the promoter region is present, and goes like this, where the terminator region is present. Means this promoter region, it is present upstream, while the terminator region, it is present the downstream region. Means 5' end of the coding strand and upstream. Now the structural gene or structural genes. Now the structural gene or structural genes is that part of the DNA which actually get transcribed. It is monocistronic in the eukaryotes, while the polycistronic in the prokaryotes. Means in case of the prokaryotes, yes, many genes are present between the promoter region and the terminator region. That's why it is the polycistronic in the prokaryotes. While in case of the eukaryotes, the structural gene, it will code, yes, for only one type of the RNA and for only one type of the protein. That's why it is monocistronic in the eukaryotes. And the last part of the transcription unit is the terminator region. It is present at, yes, it is present at which end? Yes, 3' end or 3 prime end of yes, coding strand. And it defines the termination process. Which process? Termination. At this end, the process of the transcription, it will get terminated. It will get end. Now we will see how the actual transcription process takes place. First we will see the transcription process in case of the prokaryotes. First step in the transcription process is the initiation. It takes place with the help of RNA polymerase enzyme plus sigma factor. Now this RNA polymerase enzyme and the sigma factor, they will bind to the promoter region. That is the, yes, it will bind to the promoter region and initiates the process of the transcription. Remember, in case of the replication, RNA primer initiates the process of the replication and then DNA polymerase, it brings out the complementary nucleotide. While in case of the transcription process, RNA polymerase itself with the help of the sigma factor initiates the process of transcription. In case of the replication, helicase enzyme, it breaks the hydrogen bond and two strands of the DNA gets separated. But in case of the transcription, RNA polymerase enzyme itself locally it unwinds the DNA strands. Now this RNA polymerase enzyme, it will moves along the DNA strand, it will cause the local unwinding of the DNA strand, and one of the strand it will act as a template strand. Complementary nucleotides, they will join to the nitrogen bases that are present on the template strand. Hence, newly formed RNA, yes, it will bind to the template DNA strand and the DNA RNA hybrid will be formed. Now this is the template strand of the DNA and this is the new strand of the RNA, means DNA RNA hybrid molecule will be formed in the elongation process. Next step is the termination. As this RNA polymerase, it will reaches to the terminator region, the another factor, that is the factor Rho plus RNA polymerase enzyme, they will bind to the terminator region and the RNA polymerase, it will dissociates from the DNA and it falls off. Now the RNA polymerase enzyme falls off and the newly formed RNA, it will get separated from the DNA RNA hybrid molecule. In this sequence, the process of the transcription takes place and newly formed RNA, it will be formed. Now student, in the transcription process, in case of the prokaryotes, this mRNA or this RNA formed, it is the mature RNA and it do not require further processing. Only single RNA polymerase enzyme is responsible for all three types of the RNA formation in case of the prokaryotes. And transcription process as well as translation process in case of the prokaryote, it takes place in the same compartment, that is in the cytoplasm. Suppose a bacterial cell hai, in this bacterial cell, yes, DNA say, yes, from DNA, yes, there is formation of the RNA. This process is called as the transcription and it takes place in the cytoplasm in case of the prokaryotes. And from this RNA, proteins will be formed. The process is called as the translation. Both transcription and translation process takes place in the cytoplasm in case of the prokaryotes. That's why transcription process as well as translation process, they may take place simultaneously. Means before transcription process is get completed, this translation process in case of prokaryote, it get starts. This is not in case of the eukaryotes. In eukaryotes, this RNA, it is not fully matured. Hence, there is processing on the RNA. So transcription and translation do not take place simultaneously in case of the eukaryotes, while it takes place in the prokaryotes. Now listen carefully. What we have seen in the transcription process is, the transcription process, it is the formation of the RNA from the DNA with the help of RNA polymerase enzyme. Where it takes place, in case of the prokaryotes, it takes place in the cytoplasm, while in case of the prokaryotes, it takes place, while in case of the eukaryotes, it takes place in the nucleus. When it takes place in the G1 and G2 phase of the cell cycle. The process or the steps, that is initiation, elongation and termination, are same in both prokaryotes as well as in the eukaryotes. The difference is, the in case of the prokaryotes is the only single enzyme, that is the RNA polymerase enzyme, while in case of eukaryotes there are mainly three types of enzymes, that is RNA polymerase 1 for the ribosomal RNA, RNA polymerase 2 for the messenger RNA, RNA polymerase 3 for the transfer RNA. And then we have seen the transcription unit. It consists of promoter region, structural gene and the terminator region. Promoter region, it is present at the 5' end or 5' end of the coding strand. Structural genes, these are mainly monocystonic in eukaryotes while polycystonic in the prokaryotes, while the terminator region, it is present at the 3' end of the coding strand. Conventionally, the polarity of the coding strand is considered. And then we have seen the initiation, elongation and the termination. Initiation, RNA polymerase plus sigma factor binds to the promoter region. Elongation, RNA polymerase, as the RNA polymerase moves on the DNA, the strands of the DNA, it will get separated. One of the strand, it will act as a template, its complementary nucleotide will be brought out, and the DNA RNA hybrid will be formed. And as the RNA polymerase, it reaches to the terminator region, rho factor binds to this RNA polymerase enzyme, and the process, it gets terminated. And the RNA polymerase, it will dissociates from the DNA strand. The enzyme RNA polymerase, it will get dissociated from the DNA strand, and the mRNA will be formed. This is the transcription process. Thank you.